Right, so David has come up from Oxford today. He's giving a talk in the infection control meeting, hospital infection meeting tomorrow in Manchester, talking about C. diff tomorrow. But today he's going to tell us about the interesting problem that you had in your occasion with Candida aureus. And as you can see, he's duly trained in infectious diseases and microbiology and working in Oxford. And we're very happy to have you here. Thank you very much for coming. We look forward to what you have to say.
Great. Well, thank you very much for having me. So I guess I should say up front, yeah, I'm duly trained in ID and micro, but not really someone who knows lots about mycology. So you can be kind when you think of some hard questions to ask me. But slides to advance it. There we go. So just to say a little bit about Candida aureus to start with. So this is an organism that's really come to people's attention in the last few years. First identified in Japan in 2009, and at that point, it was this was just people being diligent, really, not and noting this thing that hadn't been identified before. But then, if we fast forward to the current situation, so this is a map from the CDC as of the end of April. You've got all the countries in blue there where there have been outbreaks, and it's a particular interest really for two main reasons. One, it's been associated with high rates of resistance to treatment, and the second thing is it's particularly affected vulnerable patients in high settings. And if we look at that map and think about sort of sources for UK cases, particularly the Gulf region and Kenya and also South Asia have been sources of infections when it comes to the UK. So just some more maps here really showing you that this has been in Europe, particularly a problem in the UK and Spain, but there's also a significant problem in northeastern USA as well. And what's interesting about it is, although it's described as a relatively new problem, actually if we look at its genetics, we can see actually that it follows this quite structured pattern. So looking at the phylogenetic tree on the side there, you've got different countries that Candida aureus isolates have been taken from, and they cluster on the phylogenetic tree, and they're tens of thousands of SNPs different. So the kind of evolutionary time separating these is probably hundreds of thousands of years. So Candida aureus has been sitting quite happily independently in bits of Southern Asia or in bits of South Africa, not really mixing with Candida aureus from anywhere else. And then all of a sudden you get simultaneous reports of this causing a problem for patients in multiple places. And lots of speculation about why this is the case, but not actually completely clear. You might say, oh, it's because people couldn't diagnose it properly, and I'll come on to that in a moment, but actually searching back through an archive of Candida aureus isolates, sort of 15,000 of those going back to 2004 actually identified relatively few Candida aureus isolates. It does seem that it's a new problem. The other thing to say is it does require specialized laboratory methods for identifying it. So certain methods, for example, based on biochemical tests, either standalone or as part of commercial platforms, will tend to misidentify Candida aureus, particularly as Hemoloniae. But actually, with appropriate databases, both the main two multi-top platforms will identify it fine. But it is important that you have the right database, else you may misidentify. So. And then the sort of final bit about it is, well, what do you do about it? So the particular patients it's affecting are people who are very sick anyway, in some series that can all-cause mortality in patients who develop invasive infections, described as high as 50%. But bear in mind that the people who are getting infected in the first place are very unwell. And then from an infection control perspective, most of the advice, and this is just a summary of this Public Health England advice, it's been based on extrapolation from other organisms at the hospital. So screening people who've come from places that may be at high risk, so that's places where they're known to be cases either in the UK or from overseas. And then most of the care for patients is around sort of contact precautions enhanced environmental cleaning. So that's the background, and then what happens for Candida aureus in the UK? When does it start being a problem? And the first real awareness of this is when, in the summer of 2016, a large outbreak is described at the Brompton Hospital, and you can see some of the numbers I've pulled out there. So we've got nearly half of patients developing some form of invasive infection, and about a fifth of patients developing candidemia, and some typing that was done by Public Health England showing that these strains from the same geographical region. And so what this did was to alert us to the problem. In the US, the CDC and Public Health England at a really quite similar time put out an alert. And in Oxford, actually, we'd been happily identifying Candida aureus for some time, sending it off to the reference lab and saying this is a bit odd, and not really kind of appreciating the significance of what was going on. But then, when this alert was put out, we went back and actually looked systematically back over our cases, and we found that we'd had 4 patients who'd had an isolate which represented colonisation, and then 5 with invasive infection. And what was really striking was actually 8 of the 9 patients had been on our neurosciences intensive care unit before they were diagnosed. So this caused a degree of anxiety, and actually, while this was going on, so sort of building up to this, we had a patient who was transferred to our one of our regional DGHs, Wexham Park, where the patient was noted to have a Candida aureus in their urine following discharge from our neuroscience ICU. And then we had another case in October of 2016 where we had a bloodstream infection. And in talking to Public Health England about this, as we'd sent off the isolates, they'd been typing them through a couple of mechanisms, and could tell us that all of our isolates that we had belonged to the same strain. So this triggered a really quite substantial patient and environmental screening program from October 2016 onwards. Just to tell you a little bit about our neurosciences ICU, so it supports our neurosurgical unit and the trauma unit. There's also some plastics and ENT patients who come there. We've got about 650 admissions a year, and it's a sort of big barn-style configuration, big open space. We've got 3 side rooms at the bottom there, but the rest of it is open plan. And the other thing is that, for whatever reason, the offices at the opposite end to the entrance, and so people would quite happily be walking past all the patients to get to the office space and back again. So I'll give you the whole story to start with, rather than giving you 2 drips and drabs, but you can see that we've got this epi curve here where we had 76 patients overall who were colonised or infected. And of those patients, 72 of them were admitted to our neuro ICU before diagnosis, usually for around a week or so before they became screen positive. And contrary to what I told you about at the start, with 50% of patients getting invasive infections dying, actually only 7 of our patients got an invasive infection, and these were infections in the kind of sort of sites you'd imagine in neurosurgical patients, so line-associated bloodstream infections, and neurosurgical device-associated meningitis, mostly patients with EVDs in. And then we had one patient who actually we never captured the isolate for, who kind of came very early on, who was in green in October 2015, who was had an orthopedic device infection. We don't know actually whether that was related or completely unrelated to any of the rest of it. And then only one of the patients of the 76 died, or one of the patients of the 7 with an invasive infection died, but that was over 200 days since the last invasive isolate. So we don't think we had any attributable mortality. And when we appreciated there was a problem, we started some fairly intensive patient screening. What we did was initially screen patients on admission and then once a week, but we're actually finding that many of our screens were coming back positive. We felt we had to kind of improve the turnaround time, so actually went to screening patients 3 times a week. What we were doing there was simply actually plating screens out on some SAV agar and then identifying Candida aureus by multi. We weren't doing any enrichment, but still getting sort of pickup rates of around 9% of our screens being positive. And an acquisition rate, which we could then work out as being about 3 acquisitions for every 100 inpatient days at Woodiscombe ICU. Just to tell you a little bit about what our Candida aureus looks like, various patterns of resistance have been described. What we saw was fairly widespread azole resistance. There aren't necessarily universally agreed breakpoints for Candida aureus, so this is using Candida albicans breakpoints. So you can see most of our isolates resistant to 3 azoles there, amphotericin resistance of uncommon but present, and then no resistance to the kind of candidates. And so from our perspective, actually, that meant that many of our patients we could treat with microscopic mouth being too much of a problem. What did we do about it? Well, we tried an awful lot of things to try and get control of this, because this was actually really very disruptive. We were getting lots and lots of new cases, particularly at the main point of the outbreak. So we had the patient screening, which I told you about. We talked about environmental screening that we did. Started by trying to isolate patients, but quite quickly it became apparent that we were going to have to cohort patients, because at times actually we had as much as half of the unit occupied by patients who were colonised. And then we were also quite careful to give specific advice. Lots of our patients were either being discharged to other ICUs in the region or to the adjacent ward. And so making sure that people had appropriate advice on looking after patients when they received them. And then worked really quite hard on trying to clean the environment, but also removing as much clutter and other stuff from around the bedside as possible. So we just really minimise what was around bedsides, and then at the end, when patients were discharged, anything that was around the immediate vicinity of the patient was actually getting thrown away, and a whole sort of new bunch of things brought in. Worked really hard with the staff, and just showed that map of people coming through the unit. So we were sort of trying to avoid traffic, but also trying actually just to support people, because actually people found it quite an upsetting time, because actually they felt we're doing a lot to try and look after patients, and actually they're all getting sick, and is it our fault? What is it we're doing? And no one knew quite how it was spreading around, but there was this sense that, well, is it something we're doing and we don't know quite what? What we also did was to institute, for patients who we knew were colonised, give them a single dose of microfungin prophylaxis before surgical procedures, and having done that, we didn't see any invasive infections after having started doing that. And then there are also lots and lots of meetings, talking to people within the hospital, within the local Public Health England infrastructure, and also nationally as well. And then also trying to share experiences with other trusts that had large outbreaks. So the outbreak at the Brompton, and then a large outbreak at King's as well. So we thought, well, where's it coming from? We weren't sure at all. So in collaboration with the public health team at Portland Downs, we did quite a bit of environmental screening, and talking to them, one of the first suggestions that they made was to say, well, think about things that are touched a lot, high touch points, and also reusable multi-use equipment. And we did environmental screening using a range of different techniques, because there isn't really an established kind of, this is absolutely how you must do it. But going for things like pulse oximeters, blood pressure cuffs, equipment that came to bedside, bed rails, computers, the environment around, and even at one point actually taking apart the ventilation system, is there a problem there? And also a bit of work with active air sampling as well. And because of work showing that CPE transmission may involve wastewater, we also looked in our sinks as well, but didn't find it there. So air sampling, a couple of pictures of air samplers there. When we did active air sampling, we really didn't find a lot. We also put a whole load of settle plates out, but again didn't find a lot of Candida aureus. We found that there was one settle plate that was positive, and that had been underneath the bed of a patient who'd been transferred from one bed space into that bed space. Had the whole sort of transfer happen above them, and we just got, and they were heavily colonised with Candida aureus, but we just got a single Candida aureus colony. So air-based transmission didn't really seem to be that important. I think that just really summarises in a bit of detail what I just said. So we didn't really find Candida aureus. So it was then a question about, well, where else could we look? So we took lots and lots of samples and continued to have new cases throughout this and didn't really find anything. Everyone was quite anxious as to, well, will we ever get rid of this, or is it just the new normal? This is going to be here forever. And then one of our nurses said, well, what about these temperature probes? So these are not thermometers as you imagine, just sort of a tympanic thermometer, but actually they were rubber probes that sat in the patient's axilla to monitor their temperature continuously, because we were sort of worried about whether patients get hot or cold in terms of protecting them from brain injury. So these sat in patients' axillas continuously during their stay. And when we took one of these probes, there's a picture of the probe, and just lay it on a plate there, you can see the profuse growth of Candida aureus we got from it. So this was really quite unexpected, given we really struggled to find Candida aureus anywhere else. This was quite different. And so what we then did was to go back and take all the temperature probes that were in use. There were 5 of them being used and 5 that were broken and just not being used sitting in the cupboard. Culture all those, and actually 4 out of 5 that were in regular use we drew Candida aureus from. And then we did some more environmental sampling, having taken away the temperature probes and didn't find it anywhere else. So this is scanning EM, just to orientate you for size. At the bottom there, you can see a little gram-negative rod in the middle of the bottom, and then we've got Candida aureus that we could see on the surface of the temperature probes there. So what do? Just make sure these temperature probes could never be used again. Took them all out of use, and actually that turned out to be a really, really very important intervention. So we'd got fairly strong evidence from the cultures that the probes were important, but we wanted to try and see, well, was there some way we could triangulate a bit better that the probes were really the thing that mattered. So what we did was to take the electronic healthcare data that we've got, we can pull that into a research database, and then ask the question, well, what made some patients more likely than others to get infected? And to do this, so we set up a sort of classical case control study, so our cases, any patient admitted to the neuro ICU who then subsequently had a positive Candida aureus test, so either a screening test or an invasive isolate. And then our controls had to have come to the neuro ICU and had to have had at least 1 negative screen, and obviously have had Candida aureus isolate. And so we've got this database, which was set up as part of our sort of local biomedical research centre, and from that we could pull out relatively easily the dates and times of admissions to various hospital locations, and then we had information on vital signs, blood tests, procedures, microbiology tests, prescribing. In theory, it's great. Actually, in reality, what did we also go and do? We went through the handwritten books of admissions and discharges to the unit just to make sure we hadn't missed anyone, and to try and triangulate that we definitely had the data set right. So case control study, electronic healthcare data, and then in terms of statistics, just doing some regression modelling. And what we did was to think about exposures in the 90 days prior to the first positive isolate cases or the last negative screen for our controls. And we get a number of things. So length of stay matters. If you stay a relatively short time on the unit, then your risk increases. So high up on the plot there is increased risk. And then patients who stay a really long time have much lower risk. This is a sort of slightly strange relationship, but it may reflect that actually some of our long stay patients have subtly different pathology. They're often patients with refractory seizures or things like that, which may not involve the same amount of say neurosurgical intervention that some of our shorter stay patients have. So having control for patients' blood tests and having control for patients' vital signs, why they came into the unit, and for various procedures as well, 2 things came out. So auxiliary temperature monitoring. So 86% of our cases have experienced this compared to just 34% of the controls. So that made our people who had monitoring like this nearly 7 times more likely to be colonized with Candida aureus. Antifungal exposure was also came out with a big odds ratio, but actually the number of patients receiving any antifungal on the unit was fairly So here's a list of pretty much all the things we searched for. So it's pulled out the temperature monitoring, length of stay and antifungal exposure for you there. There was some subtle relationships with patients with lower albumin being an increased risk, those that had higher sodium values being an increased risk. A slightly odd relationship with neutrophils, where you had most greatest risk with moderately elevated counts. And then, although it's sort of slightly disappointing it didn't meet the magic P equals 0.05, temperature actually, so this is the average temperature of patients while they're on the unit associated with risk, which is quite a nice story maybe, because actually Candida aureus grows preferentially at 42 degrees in the lab. So we just wondered whether it was our, because again was something that sort of anecdotally the nurses kept saying to us, it's the hot patients that get it, which is interesting. And then a whole load of other stuff that we looked for that didn't make any difference. So it becomes an emergency where you had a central line, how long you were ventilated, spectrum antibiotics, those that didn't make it to the final. So that was another bit of evidence that these temperature probes are really important, but we could sort of try and understand a few other things as well from this data. So when we had a positive test, we continued to screen patients because we wanted to know how long patients needed to be isolated for. Did we see them actually losing colonization? But we also got a few other things from it, which I'll tell you about first. So here are all our patients plotted out, and the little coloured crosses are when they had a positive test. If you ignore the middle line of this table here that says another for a moment, and just look at the proportion of patients that were colonized in the axilla and then multiple sites subsequently versus those that were colonized anywhere else, there appeared to be the preponderance that patients also were first colonized in the axilla at the first screen, more so than at any subsequent screen. So again, sort of fitting with this idea that the temperature probes were important. How long did people carry for? Well, we found that if we did 2 screens in quick succession, because we were screening people 3 times a week, actually sometimes people were a bit enthusiastic, were a bit earlier, a bit late, so we actually got screened the same day or consecutive days. And so if we assume that loss of colonization during that sort of small period of time is pretty minimal, we can calculate how sensitive a single screen is, and that was around 78% there. So if we then say, well, you need at least 2 or even 3 negative screens to convince us you've lost Candida aureus, we can then say something about how long you carry it for. And it turns out that in our patients, probably they were carrying it for up to about sort of 60 days or more on average. Do bear in mind, though, that by the time we get out to people who have been in the healthcare system that long, actually it's the minority of patients. The numbers in the table at the bottom there just show the sort of number of patients left. So then what happens over shorter periods of time appears to be that people keep Candida aureus. How many negative screens do you need to be sure it's gone? Probably 3 is pretty good. But actually we did see one patient who was discharged and then readmitted, and then once readmitted and subject to a whole load of further broad spectrum antibiotics, actually we saw the Candida aureus come back. So there's this sort of slight unanswered question about, once someone's got Candida aureus, how can you ever be sure it's gone, and what should you do about it? Some of the UK and North American guidelines differ there, with the UK being more conservative about continuing to isolate patients who have been positive in the past. So that's the kind of epidemiological stuff that we got from the hospital data. I'll just change slightly and talk about what we got from sequencing the Candida aureus. So Candida aureus, its genome consists of about 12 million base pairs of DNA, so A, C, G, and T all lined up. And what that allows us to do is, if we imagine all those blue dots there in Candida aureus that we just see down the microscope or growing on a plate, we can then sort of subdivide it. So we can say, well, okay, we can put Candida aureus into related groups. And it kind of follows from that if infection is spreading between 2 people, then the DNA of the Candida aureus should be identical or really quite similar. So we can use this as a way of tracking how infection is spreading around the unit. If person A gave it to person B, we should be able to see their Candida aureus look more similar than a third person who got it from some completely unrelated route. And practically, how do we do that? So we grow up our Candida aureus. do some DNA extraction, sequence it, and we're paying kind of the order of 50 to 100 pounds a sample to do sort of short read sequencing, which gives us little fragments of DNA. So I said you've got a genome which is 12 million ACGs long. Each of these fragments is say 150 to 250 base pairs long. So we then have to sort of piece back together all these little fragments to work out what's going on. What we can also do is do some other type of sequencing, which is called long read sequencing, which instead of the fragments being 150 or 250 long, they're thousands long. So this helps us get some of the structure of the genome better sorted. So we can sort of use those two technologies together, and then that gives us some sort of output. So what do we get? Well, we didn't save all the isolates as we went along because it didn't really become apparent what a big problem this was until partway through. So although we had 76 patients and many more isolates, actually in the end we got 79 that were saved from 43 of the patients. We also had our environmental isolates, so those were from the temperature probes, also from a hoist, and then we had a couple of isolates as well from regional surveillance done by public health. And what I've done there is to plot out, if you like, the genetic relationships of previously sequenced Candida aureus from around the world, and to show you that our Oxford isolates in blue look pretty much identical to the light blue ones, which were from South Africa. And so we can be fairly confident that all of our Candida aureus looks like it's come from the same location. We can do slightly more than that because actually sort of slightly hidden at the top there, but we can work out from the samples we collected during the outbreak how quickly Candida aureus evolves. And we worked the answer out as about 6 mutations a year. And so we can then work out, well, if we kind of trace back the ancestry of all of our strains, when was the sort of common ancestor of all of them? So, and we can say that actually the outbreak was likely the result of a single introduction to Oxford sometime around 2012, 2013. Which is quite a long time before we had any idea that there was an issue going on, which is quite telling. This South African lineage has not been what's caused the other big outbreaks in the UK. They've been from other parts of the world, but it is responsible for probably the biggest outbreak anywhere in the world in Valencia in Spain. They've got over 300 cases in a hospital there. So this is quite a complicated tree, and the details are not really important, but what we're looking for here is related things sit quite closely together. So what you might expect is if a particular thing is the source for another infection, they'd be close. And so what we see is we find that patients, when we take multiple different colonies, are often colonized with different strains from the outbreak. And then also the temperature probes have multiple strains on them as well. So just running the sort of sequences from one temperature probe there that appears in 2 different places. And what we find is that actually all those blue and green circles there represent where we found the temperature probes in this tree. So it wasn't that our temperature probes were just a tiny bit of the outbreak. They seemed to represent everything that was going on, and they were closely mixed up with all the different patient samples. Taking you back to our sort of layout of the unit, what we did was to say, well, if you kind of went round it clockwise or anti-clockwise and you said, well, who's in the next door bed to me? If they have Candida aureus, are they more likely to be genetically related than someone on the opposite side of the ward? And actually it turns out there was no relationship at all between kind of where you were on the unit and how likely you were to be closely genetically related to another case. So whatever was spreading Candida aureus around wasn't kind of spreading from one patient to the next door patient, it was spreading as effectively across the unit to different places as it was to the next door person. So again, really pointing to the fact that it probably wasn't sort of something spreading through the air or direct transmission between patients. And I guess the nicest bit of evidence for the temperature probes being important is what happened when we got rid of them. So plotted for you there when we removed them. We didn't initially completely see the Candida aureus go away, but it did after a period of time. And actually, we've not had any new Candida aureus in Oxford since November 2017. So the number of opportunities I'm going to get to talk about Candida aureus or to do work on Candida aureus is fast diminishing because actually it's gone away completely from our hospital. That's not to say that it couldn't obviously be reintroduced. It perhaps is interesting just to talk you through the kind of tail end of the outbreak. So what happened was, having had all these stuff scattered all around this different tree, when we got to the tail end of the outbreak, actually many of the things were actually all just tightly genetically clustered. And if we plot in the kind of line graph there, when patients stayed in the unit, we have this gap, which I've flagged for you in orange, which shows we sort of had nobody on the unit who was Candida aureus colonized, and yet pretty much genetically indistinguishable Candida aureus came back a month or two later. We never found how it did, but a clear signal that there was something about something in the ICU that allowed it to persist. And I think this is essentially just showing very similar. So just to finish with, we could ask the question, well, why did the temperature probes spread the outbreak? I don't know what you would clean bits of reusable equipment within your hospitals, probably something like these wipes or similar. And that was certainly fairly well established customary practice in the unit. The manufacturer of these probes recommended something completely different, which involved immersing them in a solution for more than 10 minutes and doing various other things to them. But in reality, that just wasn't practicable. And it hadn't been a problem. At least you can tell they look fairly old. I think these are sort of 10 or 15 years old, and they've not caused a problem to date. But actually, if we kind of zoom in at the end of the probe there, you can kind of see that around the metal bit, you've got this rubber sheet, but it's got this gap underneath it, which is really difficult, if not impossible to clean effectively. So there's definitely something about the structure of these probes that promoted persistence of Candida aureus. The other thing, though, that we didn't know at the time, but these wipes contain quaternary ammonium compounds as the way of disinfecting. And actually, if you look at how active those compounds are against Candida aureus, so what this plot here shows is a number of different Candida species and then MRSA on the right-hand side, and various different disinfectants that can be used. So you've got various bleach-based disinfectants to start with along the plot, and then in the middle, the hydrogen peroxide-based things, and then over on the right-hand side, you've got the things that are based on quaternary compounds. And the bar you want to look at is the one with the sort of diagonal shading, so the third bar along. And you can see that essentially the extent of the reduction in Candida aureus seen with those is only about one order of magnitude compared to some of the other disinfectants, 6 orders of magnitude. So not only have we got something that was hard to clean physically, but actually what we were disinfecting with was likely not very effective anyway. And then just coming up to that sort of last point I made about Candida aureus being able to persist in the environment, this is where people have deliberately contaminated a piece of plastic with Candida aureus, left it, gone back to it and tried to recover Candida aureus, and done that in a couple of ways, looked for viable colonies or looked for biochemical evidence that Candida is there. And certainly it could be recovered up to 14 days or even longer after it's been left in the environment. So it's got quite good ability compared to some of the other yeast to persist on plastic healthcare surfaces. So I'll just sort of finish there and say that actually this sort of one of the largest outbreaks that's been described anywhere worldwide. Happily, sort of a good outcome in that we've not had anyone that's been colonised or infected since November 2017. And persistence on reusable equipment drove the outbreak. And this has relevance to Candida aureus, but I guess it's got relevance more broadly to thinking about infection spreading in the healthcare environment, just to think about the role that equipment might be playing, particularly usable. So just finish there by thanking lots of the people who did most of the work. And,
So any thoughts or questions about that? I have a couple.
David, I think it's interesting that now this is a resistant yeast. You didn't find any correlation with previous antifungal use. There were only 3 patients who had been on fluconazole previously, and It's quite remarkable.
I think that partly just represents it's the sorts of patients that are coming into the unit. Actually, many of them don't have a lot of prior healthcare exposure. They often come in because of a head injury or something like hemorrhage or something like that. So they've not had, they've not been exposed to it. As you say, actually, only a minority of our patients are exposed to it.
Any questions from across the city? We can see you, Elaine, there.
Well, I mean, I'm not a clinician. I should first, I think, preface any questions that I've got with that disclaimer. But I think it's quite interesting that this particular bug is resistant to the types of disinfectants that you would normally use. Have you looked at means of reducing contamination going forwards? Do you have better ways of decontaminating equipment than what you've been previously using?
Yeah, so I think it's really interesting. So actually, in terms of the temperature probes, we just got rid of them completely and now measure people's temperature differently. In terms of what we used for environmental cleaning, we went to using things with sort of high availability of chlorine, which appears to be the most active thing. I guess there's a broader point that infection control isn't a static thing. It's a sort of evolving balance. We thought we had it right because we were controlling everything with what we had in place. And then you get a new organism coming along and it actually tips that balance, and you have to think again about what you use. And so we ended up doing a lot more bespoke cleaning of things, using a lot more single-use equipment, because actually the commercially available products didn't necessarily offer what we needed.
And in the end, are you able to deduce anything about whether or not this outbreak? I mean, I know that you've shown that there was non-relatedness between these isolates in terms of genomes, etc. So, in terms of explaining what happened, are we looking at a situation where a single isolate came in and was then retained on equipment, and then something else entered into the environment? And is it standard practice at all in any walk of medical microbiology practice in hospitals at the moment to just screen the incoming patient population for these types of organisms?
So for things that have been common, that has been part of people's sort of practice. So for example, thinking of things like MRSA, people being screened on admission to ICU for that would be fairly commonplace throughout many healthcare systems, including in England. And similarly, screening for patients at risk of very drug-resistant gram-negative bacteria would also be done on a sort of risk basis if people had come from healthcare overseas. When this started, there was no real appreciation that we should be thinking about that for Candida aureus. But I think now, if a patient did come from or had been staying in a hospital in the healthcare system where Candida aureus was a problem, then the advice would be that patients should be screened. The other bit of advice in screening would be is if you find a case that you weren't previously aware of, that should trigger screening of patients around them to try and sort of contain further spread before it happens. Because actually, once this becomes endemic, the experience has been for hospitals in the USA and the hospital in Spain and Valencia that this has been really, really difficult to get rid of once it becomes set up. So a lot of focus on trying to detect early, try and sort of get control early and not allow it to establish itself.
Any other questions? In South Africa, I learned that the Candida aureus is now as frequent a cause of candidemia as any other pathogen. And yet you had very few candidemias overall. And maybe that's because of the patient population, because they're not critically ill, big surgical, sort of abdominal surgery patients, lots of antibiotics. Do you think that's, so the risk factors of candidemia are sort of not so overwhelming in your patients, but they are probably in South Africa?
Yeah, I think probably the risk factors for bloodstream infection with Candida aureus is probably very like some of the other candidas inside our population, as you say, but they may have been sick, but a lot of it was related to their sort of their brain rather than anywhere more widely. And so actually the amount of invasive Candida disease we saw overall, I mean, I didn't include that, but it didn't really change that much, and it wasn't some background rate anyway. So maybe we were fortunate that we had this problem in our neurosurgery ICU, whereas if we had it in one of our cancer surgery ICUs, that might have been a different story.
The other point is that I participated in a workshop last year with the Dutch and one of the international societies, and they think that we should be using salt-containing fungal media to screen. And the reason for that is because you then you exclude lots of the other candidas. So if you grow something on that at 42 degrees, if you want to screen at 42, but even at 37, it's very likely to be Candida aureus. You didn't do that because this was earlier in the evolution of thought.
I think we essentially just had to sort of make something up because there wasn't really any established guidance, and it's certainly possible that what we had was insensitive, but it turned out to be good enough for our use, and it allowed us to follow what was going on. We could see the signal and whether we've got control. Yes, I mean, people would advocate also doing enrichment culture as well, which we didn't do.
And you had a multitask, so you could identify them like that.
Well, what's kind of, I mean, we've been identifying them like that, beautifully sending them off since the middle of 2015 or whatever, but it's kind of interesting. It partly reflects the fact that I guess we're quite fragmented as a service. We might, one of the consultants might look after that particular part of the hospital for 1 month at a time, so actually you might get one case, which is a bit odd, but you kind of then, it's a different person the next time, a different person the next time. So actually that kind of global picture you didn't necessarily build up, without being unkind to people at public health England. I guess they were sort of building up a picture of what was going on, but it was only kind of really when the thing at Brompton came to the fore that then there was much more awareness of those.
Yes. Yeah, that's right. You did a sort of look back, didn't you? Because
identifying streets.
No, because we kept strains for 25 years, maybe 30 years. We've got them all. But you went through the hemaloniais and things that have been identified by.
You identified the hemaloniums by
yeah, or earlier in the era.
But originally by
32C and things like that, API and things like that.
Yeah.
So I think.
I think that's the real puzzle about it is where's this come from? Why has it come?
Yes.
Lots of people are prepared to speculate about, is it something that we're doing different in healthcare? Is it something that's changed in the environment? Is it to do with azole use in the community? But it seems a little bit difficult to buy that. If you look at the place on the map where it's come from, you get sort of over South Africa or India, and then you look at azole use where it happens in the world. It isn't necessarily centered on in quite the same way. So
I know that in Japan they're still seeing isolates, but they're ear isolates. They're localized infection, external otitis and stuff like that. They really don't cause Candida in much the same way, which is a bit peculiar. So maybe I think there may be differences in the clades in terms of transmission and risk. So.
Probably true. It probably is just this being labelled as a Japanese fungus in the media is a bit unkind because it really just reflects their diligence rather than any kind of
that's right.
And spreading it around the world.
And actually, it was first in Korea, wasn't it?
Yes.
We just didn't know that at the time.
Yeah.
But it is extraordinary, isn't it, that you've got essentially a completely new fungus that's appeared in a decade and it's gone with multiple different clades and multi-resistant. I mean, it really is very strange, isn't it? Just imagine if that was a virus. I mean, the WHO emergency of all proportions.
And we don't really understand why.
Yeah, so I think it's really quite interesting.
Can I ask a general question about the biology of this pathogen? So, presumably from what you're saying, David, there is some knowledge about certain phenotypes that are associated with the organism, that relate to its ability to tolerate salt in growth media, et cetera, and presumably by virtue of the types of infection it's causing, it's a thermotolerant organism as well. But I just wonder how extensive any phenotyping analyses have been thus far, and if people like you, David, or have accrued collections of isolates, whether there's any scope for doing some work there that would tell us a little bit more than the nucleotide sequence of the organism would, for example.
Yeah, I think it's really interesting. It's not something that we've done. As you say, yeah, no, we do have a collection of isolates, and I guess there are global collections of isolates from different lineages that might behave differently, and so understanding the extent to which they vary phenotypically would be interesting. I think we stumbled across this by accident without necessarily having a huge background in sort of fungal biology in Oxford, and so I guess we did the very applied thing and did the thing which we knew how to do, which was sequencing. And we were lucky because it's haploid, and so we could get away with just doing exactly the same thing as we've been done for sequencing bacteria in terms of the sort of bioinformatics. So from our point of view, actually, there are a few things that just made it quite easy for us to work with. I'm sure there's more. I'm sure that there's more that could be done, and if people have got ideas for things they'd like to do, we're very open to that.
Any more questions from across the city?
Not from my end.
No? All right, that's fine.
Thanks very much.
All right, good. Well, thank you very much, David. Again, very much appreciated. Thank you for coming. It was nice to see you. And great. We look forward to, hopefully we'll get too many of these bugs through the door. I really want an outbreak. Thank you very much. Great, thanks.
Okay. Thank you.
Why didn't you